Welcome to the Applied Biosystems Taq Talk video series, where we break down real-time PCR to make it easier for you. Let's talk about some common techniques for mRNA quantification using real-time PCR. Relative quantification is the most common type of qPCR analysis and is used to measure gene expression changes in a sample relative to another reference sample. But sometimes relative expression change data are just not enough. For instance, let's say I'm looking at samples infected with HIV, and I need to know exactly how many copies of virus are present in each sample. What other options are there when you need absolute answers? Using real-time PCR, Applied Biosystems Taq gene expression assays can be used to determine the absolute quantity of specific mRNAs. Absolute quantification is the precise measurement of a target's molecular concentration. In each absolute quantification experiment, also known as standard curve analysis, samples of a known quantity are serially diluted and then amplified to generate a standard curve. The quantities of unknown samples can then be extrapolated from the standard curve. The main hurdle in absolute quantification is the generation of this standard curve. Although it seems simple in principle, there are lots of things to consider. Your standard needs to meet several criteria. First, the quantity of a sample must be known by some independent means. For this step, the concentration can be measured by a spectrophotometer and converted to the number of copies using the molecular weight of the DNA or RNA. Second, the standards should closely resemble the target from a biological standpoint, and it is very important that the DNA or RNA be single pure species. For example, when measuring gene expression of RNA transcripts, you would want to use in vitro transcribed RNA. Take care here because purity will be an important factor in the accuracy of your measurement. Third, and finally, don't forget that your excellent pipetting skills can be put to good use here. One of the major pitfalls for scientists setting up standard curves is that they do not pay enough attention to the accuracy of pipetting of technical replicants. For the best results from your standard curve, ensure that your pipettes have been recently calibrated, and be very careful when making dilutions and pipetting into the plate, and ideally, make use of low retention tips. Now that we have our standards, let's set up a dilution curve in our plate. We recommend running a triplicate with 10-fold dilutions and at least 5 data points. When set up correctly, all Applied Biosystems real-time PCR instrument software will generate a standard curve for you from these points. The equation of the linear regression line through these points is then automatically used to calculate the quantities of any unknowns on the plate in the same units. For example, in this curve, I have 5 points, starting with 20,000 copies of my target as the highest concentration, going down to 1,250 copies. The standard curve plot is showing the input on the x-axis as log x and the CT values on the y-axis. Quantities are then determined from this equation. We'll remove any outlying replicates or points when necessary. Solving for x using the CT values of our unknown samples will give us the missing quantities. Notice that they will be in the same units as our standards: copies for copies, nanograms for nanograms, and so on. So now we have determined the absolute quantities of our unknown samples. Whew, making all those standards was a bit of work. Is there any other way to get this type of data without the need for standards? Why, yes. There is one other way to get absolute quantities with Taq assays, and that is to use digital PCR. Digital PCR works by partitioning a dilute, approximately single copy sample into many individual real-time PCR reactions. A portion of these reactions will contain the target molecule and are positive wells. The other wells do not contain the target molecule and are negative wells. Following PCR analysis, the fraction of negative answers is used to generate an absolute answer for the exact number of target molecules in the sample, without referencing to standards or endogenous controls. Thanks for watching, and be sure to watch more Applied Biosystems Taq Talk videos to learn more about how to elevate your qPCR research.